So today is our last in the series, or the No Neuroanatomy series for this summer. So thank you all for everyone who's been here the whole time. If you haven't, then we'll give you the information about how to access everything that you can watch at your leisure. So my name is Sarah Sims, and we're pleased that you could all join us for this week's lecture in our 7-week neuroanatomy mini-series that brings you lectures from experts in the field covering different neuroanatomy topics each week. All right, so this series was created by trainees and early career neuropsychologists to provide free, high-quality didactic opportunities. We'd also like to recognize the planners for the No Neuroanatomy series. Right here. And we'd also like to thank our sponsors for their financial support for the series. And before we start, we'd like to make sure everyone's aware of our YouTube channel. That's where you can find all the No Neuropsychology, as well as this series, the No Neuroanatomy. And this last one will be uploaded after today. So you can like and subscribe to these lectures. So find us on YouTube. Here are disclaimers for the series. The training is not meant to replace formal education in neuropsychology, and the views of the speakers are their own. Okay. Questions can be submitted via the Q&A box on the lower left of your screen, and a recording of today's lecture will be provided on our website and YouTube channel later this week. All right. It's now my pleasure to introduce Dr. Vischer, who's joining us today to give us a lecture about neural networks. So Dr. Christina Vischer is an associate professor of neurobiology at the University of Alabama at Birmingham. She has an undergraduate degree in physics from Carleton College and switched to neuroscience for graduate school at Washington University in St. Louis. And she did her postdoc at Brandeis University and Harvard University. Her lab at UAB studies how microscale aspects of brain connections change with aging and with experience. She focuses in particular on changes due to learning to use peripheral vision after loss of central vision. The lab uses MRI tools to examine changes in brain structure and function, as well as careful psychophysical manipulations of behavior to examine function. We're so glad that you could join us today. And so go ahead and take it away with your slides.
All right. Thank you so much, Sarah. I'm really very honored to be here and to get to talk to you about networks. Well, neuroimaging and networks. I'm going to focus on the neuroimaging side of that. If you have questions, I would love to answer them, but if I miss seeing them, hopefully somebody can let me know.
Yeah, I'll go with, there's a question. Everyone's welcome to put it in the Q&A box at the bottom, and then I'll pose them to you as they come up. Good. Thanks.
Okay, awesome. We think about the brain as in a lot of different levels. And we as neuroscientists and as neuropsychologists, we're aware that all these levels exist and interact with each other. And different scientists study different levels. So for example, we can study behavior, right? We can study how small changes in stimuli give rise to changes in behavior. We can study the brain at the level of the whole system, at the level of networks, neurons, individual channels, individual molecules, like how does this particular drug interact with a channel, for example, and at the level of chemistry. So we can hit any or all of these levels with our analyses that we do. And we're going to get slightly different answers depending on what kinds of questions we ask at each of these levels. All neuropsychology is really focused on the behavior. And the neuroimaging tools that I'm going to talk about today are focused on the system or network level. We're going to be talking about macro scale connections among different parts of the brain. And we know that, for example, the visual system is hierarchically organized. There are multiple interacting networks that give rise to the visual system. We know that visual processing, for example, is modified through top-down influences. We take in information through our retinas to our visual cortex, and the information processing that happens there is modulated by top-down information. And we know that learning can change the nature of this processing. So there's this input to output connection. Right. It's also true that this sensory motor system is hierarchically organized in a similar way. Motor output is guided by sensory input, and learning can change the way that this information is modulated. We also know that the brain regions involved in memory are comprised of interacting networks. The brain regions involved with language are comprised of interacting networks. Everything we have studied so far has behaviors that are modulated by interacting networks within the brain. So I'm going to quickly talk very briefly about language, which is near and dear to many of your hearts, I know. We know a good deal about the neuropsychology of language. And how do we know that, right? We know this because of the work of people like Paul Broca and Carl Wernicke, who very carefully studied patients who had very specific problems with speech and then looked at their anatomy once they had passed away, right? It's from Broca that we've developed the idea that language, that expressive language involves the left frontal lobes. It's from Carl Wernicke that we get the idea that receptive language involves a more posterior set of regions. These are both generally on the left side. So usually these lesions that were observed by those scientists and others since, usually those lesions are due to stroke. And this kind of experiment is an example of localization of function through natural experiment. There's lots of examples of that kind of work in the literature. For example, hemi-neglect in the attention literature, where people who have strokes in the parietal lobe have problems attending to different locations in space, or one half of space. Okay, so the new information that we got from Wernicke and Broca is, one, we understood that there was localization of language to those regions on the left side, but also we understood that speech fluency and information content are processed pretty separately. And since then, there's been tons and tons and tons of literature looking at different aspects of language. So for example, this review from 2010 shows regions that are involved in not just speech fluency and information content, but pseudo words. We're comparing pseudo words to words and sentence comprehension. Different parts of this language network are important for these different kinds of tasks. So these are examples of looking at very specific parts of the brain and identifying their function. But of course, none of these single brain areas would be able to do anything alone in a dish, right? It needs connections to the rest of the brain, the rest of the body, in order to do what it does. The function of any neural circuit depends on its wiring, depends on what else it's connected to. So it's essential to understand what's connected to what in order to understand the system. So there's sort of two approaches that people have taken when looking at neuropsychology. One is the modular approach. The idea is you identify what area serves what function. So Broca's area serves the function of expressive speech. So breaking this area means that function doesn't happen any longer. But there's another approach which can be used in conjunction with this modular approach, and that's the network approach. The idea is you look at the system as these areas are connected in these ways. We can try to understand how they work together. And I like to talk about how this happens in this way. So the nervous system is sets of distinct parts. There are different neurons in different parts of the brain. We can identify, the different lobes, the different areas within the lobes. They're somewhat separable. So one of the basic questions that face systems and behavioral neuroscientists is, how do those parts work together? So if each part of the nervous system has specialized functions, I'd like you just to think about how they might work together on your own. Think about in what ways they might work together. Do they work together like a car's engine and a radiator do? So each does a very specific independent job, but both are needed to make the car go. Do they work together like words in a sentence? That is, they have their own meaning, but with the overall meaning determined by the whole. Like voices in a chorus. Each lends a distinct but not freestanding note to the harmony. Do they work together like pieces in a jigsaw puzzle where each contributes, but nothing's particularly meaningful on its own? Are they like transistors in a computer? That is, they contribute a mysterious bit to whatever task is being done. Or like molecules in a gas, you can't define the temperature of a single molecule, but once they get many of them together, you can start defining things like temperature. So I want you to think about this idea as you're going through your, learning about the different networks of the brain. Okay, so we have a lot of different network approaches that we can use to try to better understand the brain. And we can also use those same network approaches to understand other things besides the brain. And I'm sure that you have heard about some of these network approaches as applied to aspects of your life. So for example, friendship networks or connection networks of people. So LinkedIn had this thing that you could do where you could identify all the connections, all your friends on LinkedIn, and it would make a map like this one. And in this map, every little dot here represents a person. And if they're friends with each other, there's a line between the dots. And any two dots that share a lot of connections get put closer together, right? So you can see this person, I chose this person randomly off the internet, Steve Cummins, okay? This person has a lot of dots that are tightly connected over on this side, but they tend not to know the ones over here, who tend not to know the ones over here, for example, right? So you can see just from this image that this person has two major sets of friends that don't tend to know each other. And knowing what we know about human relationships, we can probably say that this person has had two major jobs in their life. And in fact, that seems to be the case. Apparently, this guy worked at Panasonic, which is all the orange ones are Panasonic, and a company called MCore. All the blue ones are from MCore, and they have family and friends and some school friends over in these other networks. So just by looking at these connection networks, we can tell a lot about this person's relationships and a little bit about their history. They've had two major jobs. Okay. What about this one? This is another person, Ali Imam, again, chosen randomly. And with this one, you can see something very similar. So this person went to Carnegie Mellon, had a lot of classmates, worked at Yahoo. They all know each other, but the Yahoo Analytics people know each other more on average, and a lot of LinkedIn friends. But with this one, we can think about which of these friends are most influential. So we can look at these connections and we can see that like this one is very influential among the other Carnegie Mellon classmates, but doesn't have a lot of connections to other networks. Whereas this one, Micah Alpern, is connected to a lot of the other friends. So this person would be able to, make a lot of connections to Ali Imam's other friends. If Ali Imam has information he wants to get to his friends, maybe he should send it to Micah. So you can tell a lot about a system based on its connection structure. This is true for friendship networks. There, the individual nodes are people. I'm going to use the term nodes. And the individual connections between them, which we call edges, are friendships. So we can play these same kinds of games with brain data. And I'm going to show you some of the results of trying to do that. So this is an image from a very influential 1994 paper by Fellman and Van Essen. And here we're defining the nodes as brain areas. So each of these little colored squares is a different area of the brain. And the edges are axons actually physically connecting these brain areas. So down here, this is the retina, the LGN, early visual cortical regions, and way up here we have the hippocampus. So we're not even including the frontal lobes here. So you can see that there are a lot of complicated connections among all these brain regions. So how do they work together? So to understand how they work together, we have to try to understand the brain as a network, not as individual nodes. Okay, so in order to do that, I'm going to tell you just a little bit about some of the methods that we use to look at these connections. All right, so we have many levels of understanding neuroscience, and I'm going to go back to the neuron here, which has action potentials that send information to other parts of a network. So action potentials are how neurons send information to other neurons. And neurons can send information to other neurons within the same network or within far away networks. And we can measure these changes in action potentials in firing between, in sharing information between these neurons. We can measure those with changes in electric field, which we measure with action potentials. And as you'll see, we also can use interesting MRI techniques to measure what's going on at the connections between neurons. Okay, what happens is when a neuron sends a signal to another neuron, that causes there to be these enzymes that produce compounds like nitrous oxide and prostaglandins that dilate blood vessels nearby. You don't have to know the details of this, but you have to know that there's a dilation of blood vessels nearby, causing an increase in the amount of oxygenated blood in that area. And when there's an increase in oxygenated blood, we can measure that using MRI. So at rest, when those neurons are not firing, when blood goes by, a little bit of the oxygen comes off of that hemoglobin. So there's hemoglobin in blood. And when it's oxygenated, I've colored it red. And when it's deoxygenated, I've colored it blue. But when we send more oxygenated blood to an area, there's just a lot more oxygenated blood than there is deoxygenated blood. And we can see the difference between those in an image like this. This is a difference image looking at where there's more oxygenated blood during a visual task in this case. Okay, so we can look at the levels of that oxygenated or deoxygenated blood, and it changes over time without any stimuli present. Your brain is always active. It's never sitting still. So for example, this is an axial view of the brain. Right here, we're looking at the motor cortex, so right about here in that person's brain. And you can see over time, the magnitude of this signal, which we're calling the blood oxygen level dependent signal, that signal changes over time. like this, without any stimuli present. And now I'm going to show you a little video. So if I were to look at every part of the brain and color it based on that bold signal, I could look at one region, the motor cortex, which would be right around here. That yellow region that I showed before would be right around here. But here I'm looking at the entirety of the dorsal aspect of the cortex. All right. So I'm going to show, this is sped up just a little bit relative to what's actually happening right now in your brain, but it's very similar in your brain right now. So this is activity in the brain. When it's more blue, there's less activity. When it's more red, there's more activity. And I want you to notice whether you notice any patterns in this activity. For example, you might notice that when this region here is active, it tends to be also active on the mirror symmetric location. There's a lot of mirror symmetry going on. So for example, here on the sensory motor strip, when it's active on one side, it tends to be active on the other side. So these patterns of activity over time are very reproducible. Well, their patterns are reproducible, but at any given moment, we can't necessarily tell where we're going to be in this pattern. Okay, so this is just a dorsal view of the human brain. Just to orient you, this is the front of the head, this is the back of the head, and the corpus callosum would be right in here, but we're not showing that. All right, so that is how bold fluctuates across the whole brain. So if I looked at this one region, again, it's fluctuating over time. And here I'm plotting the fluctuations over time of two regions that are supposed to be mirror symmetric. So two, the left and the right motor cortex. And so that's in yellow and in pink. And you'll notice, just like you noticed when you were looking at the whole brain image, that there's a lot of similarity to the time points that our left and right motor cortex tend to be active, right? We can predict what the left motor cortex is doing just by looking at what the right motor cortex is doing. And we define that as having a high Pearson's correlation. And it just so happens that mirror symmetric regions, especially the motor cortex, have a really high Pearson's correlation. That was shown by Biswal in the 90s. So what does that mean? We're going to talk about some vocabulary first. We refer to two regions that have really strong connection strength or correlation strength like this. We're going to refer to that as being strongly functionally connected. Oops. So these two regions that are changing in concert, we're going to call them strongly functionally connected. So that's a correlation of the MRI signal between any two regions. I want you to remember that this is an indication that they share information. It's not necessarily causal. So if the left and right motor cortex are both rising and falling in concert, it doesn't necessarily mean that there's information from the left cortex that's moving to the right cortex. For example, this here is a plot over time, a year, of sales of ice cream in dollars and forest fires in number of acres. And so you can see that forest fires are highest in late summer, but also ice cream sales, they're also highest in late summer. So does that mean that ice cream sales cause forest fires? I mean, you all know it, that it doesn't, right? It just means that they're both driven by heat. They tend to be driven by the heat of the summer. So keep in mind, when we're looking at functional connectivity, we really mean that there are correlations among two regions. And that could be driven by a couple of things. One is sending information from A to B, or from B to A, or from some third region, C, could be driving both of them. Like, for example, the heat of the summer driving both ice cream sales and forest fires. So a set of brain areas that are functionally connected to one another is sometimes called a network. Right. So we can, for example, plot, instead of just plotting two regions, I could use this left motor cortex region, and I could look at how every single part of the brain is functionally connected, is strongly connected to every other part of the brain. Not just this motor cortex, but also this region and this region and this region. And I could say, I could color that part of the brain like bright yellow if it's strongly functionally connected, and color it another color if it's weakly functionally connected. And that's what Sarah Nolan did. And she's on this call. She's the person who introduced. So I'm showing some of her data here. So the brain regions that are shown in bright yellow here are regions that have strong functional connectivity to this region back here in the posterior cingulate cortex. And you can see that there are a discrete set of brain regions, and these tend to be called the default mode network. And I'm not going to talk much more about that, but if you want to know more, that's a keyword you can use in Google Scholar. So we put, Sarah put another seed in the dorsal anterior cingulate right here, looked at what parts of the brain are strongly functionally connected to that region. And this is what we see. Very different looking map. And when you make those kinds of maps, we can see that they tend to be pretty segregated. So different regions, different networks, they fit together like a puzzle pieces, right? They're not overlapping very much. So we call this segregation. So the degree of this segregation can be measured in several ways and seems to relate to behavior. And sort of as an aside, this data came from only people who are over 85. It recapitulates what's been seen in the younger data too. I'm just showing you these data that Sarah made from people who are in our oldest old group. All right. So these networks are super reproducible. We see them in older adults. We see them in younger adults. We see them in virtually every kind of study that's ever been done. I'm just showing you a couple of examples of the types of analysis strategies people can use. If you analyze your data so that you use every single point in the whole brain image in order to get your networks, you get very separable networks. If you just use a couple of different points, you get very separable networks. It doesn't matter how you slice it, you get the same thing. So I'm going to continue with vocabulary. So another vocabulary term is the term region of interest. By that, we mean a group of contiguous voxels. Voxels are like volume pixels, so the regions in the individual spots in the brain. So these are just an example of someone's regions of interest. So you can see here in this subcortical network, we've got one yellow region of interest. Here we might have this red region of interest. Here we have this red region of interest here, which is a different brain region. So each of these would be regions of interest. Okay. Continuing with the vocabulary, we can describe a correlation matrix. The idea behind a correlation matrix is to answer the question, who is connected to whom? So if we think back to that LinkedIn network, we have, 5 people here, A, B, C, D, E, and this is how they're connected. We don't have to draw that complicated image that you saw on that LinkedIn page. We can instead just make this matrix. So A is connected to B, but not to E. So we put a 1 here and a 0 here. And you can see that this is mirror symmetric, because if A is connected to E, E is also connected to A. So this is called a correlation matrix, or sometimes a connectivity matrix. And this is a more complex version of exactly the same thing. So each of these lines here corresponds to one region of interest, just like these. In fact, this is for these region of interests that I showed you earlier. So each of these columns or rows is one region of interest. This is mirror symmetric along the diagonal, because if you're connected, if the right STG is connected to the hippocampus with, a mid-level connectivity, then the hippocampus is connected to the right STG with the same level of connectivity. It's completely mirror symmetric. Okay, that is a correlation matrix. Just an easy way to describe these networks. Okay, and most of the math that we're going to end up doing on in order to describe this connectivity is done on these correlation matrices. And here's just another representation of those correlation matrices with some example regions of interest on this brain here. Another set of vocabulary words is resting state functional connectivity. That just means functional connectivity measured while the subject was resting. So in this image that I showed you, the subject is just sitting in the scanner, not doing anything else, resting. But we could have had the subject be, watching a movie or, playing a little video game. And that would be, we could do exactly the same measurements on data from that subject while they were playing the video game. We wouldn't call it resting state functional connectivity in that case. Okay, so we can measure functional connectivity in any task we want. All right, so what types of analyses can you use resting state functional connectivity, as it's sometimes abbreviated in? We can look at region-wise analyses, sometimes described as correlation matrices. So I just showed you these correlation matrices. Each of these columns or rows would represent one region of interest, and the colors represent the strength of correlation at each of these regions of interest. Also, you can look at voxel-wise connections. For example, this whole brain image that I showed you here. And when you do usually these region-wise analyses, you can look at the structure of the network that arises. You can look at patterns of connectivity among these brain regions, and that tells us something about how they process information. Just like we talked about with your LinkedIn friends, we can tell something about how each of these networks is going to work. So you can tell whether information can run through the network in a serial way versus a parallel way. So for example, this cluster tree type of network is going to have information flow serially, whereas this mesh network, you could have information flow from left to right in a number of different pathways. So you could do it in parallel. What happens when a particular node goes out in this star network? Nothing is going to be connected to anything else in that case. So this is very susceptible to problems of this node going out, whereas that's not so true in the mesh network. So you can think about these things. Yeah, so these networks, these kinds of networks are just smaller, simpler versions of these kinds of networks, these brain networks that we've been defining. So the question is, one question is, how does damage to specific nodes in these brain networks, how does that damage performance? So, for example, this is another representation of the same general idea. Each of these circles is a brain area. Each of the lines is a pretty strong functional connection. And the circles that are bigger have more friends, basically. They have more functional connections. And so this is representing what's happening in a healthy control. So what happens if we take out this node here? Maybe a node that doesn't have a lot of connections. And how would that be different than if we took out this node here, which has a lot of different functional connections and is interacting with a whole lot of other brain regions? That's a similar question to what we were asking here. We asked who are the most influential friends, right? If Ali got mad at Priscilla and just stopped talking to Priscilla, right? Then it wouldn't have an enormous effect on his networks here. Whereas if he severed all connections to Micah, it might have a more strong influence on his day-to-day life. So we can ask that same question: which networks, or which nodes rather of the network, are more, quote, important in human neuropsych data? And this is work done by Warren et al. In 2014. What they did is they defined these networks in a group of healthy controls, and a group of people who have had stroke or other, usually stroke damage, a lesion, to regions that in the healthy controls had been defined as low connectors. So kind of like Priscilla in this image. So not central to the whole network. Oops. So low connectors damage. And then we had a few or participants who had damage to high connector nodes. So they have damage to some of these nodes that are very strongly connected to other regions in healthy controls. And so what they did is they measured a whole lot of different neuropsychological tasks, right? In domains of orientation, perception, memory, verbal function, conceptual form, executive function, etc. And here, the impairment of a participant is indicated by the coloring here. So if it's a very intense color, that means they're very impaired. And so all of the patients, this is 30 patients, all of these patients have had roughly similar amounts of damage in their brain. But the ones that are labeled control have lower, have less, low connector hub damage to lower connector, or damage to nodes that aren't as strongly connected to the others. Whereas these have damage to nodes that are more strongly connected to others. And so you can see that these patients, yeah, they have problems, right? Patient 7 really has a problem with perception. Patient 2 has a very strong problem with adaptive functioning. But generally, their problems tend to be pretty discrete, not entirely discrete, but they're not having wide-scale problems. Whereas those subjects who had damage to a connector hub have wide scale problems. They tend to be impaired in a larger number of domains than the controls. So this is telling us, again, that the network itself is important, and that the elements of the network that are connected are really strongly connected to other elements of the network. Those elements are going to be important for a large number of domains of processing. So in summary, I've told you that networks are essential for neuropsychology. Scientists can identify the function and structure of networks individually. We've talked about the language network. You guys have studied tons of different kinds of networks. We've got the default mode network, the frontoparietal network, the vision network, the motor network. We understand a lot about the function and structure of those networks. And we can identify how networks work together and how they're disrupted selectively in disease. So that's what I wanted to tell you about, and I would love to take any questions that you have.
Hi, am I taking a second? There's my video. Okay. Hi, thank you so much for all that. That was great. So I do actually have a few questions that I would like to pose. And anybody who is interested in asking some questions, you can drop those in the Q&A box for us. So my first question has to do with the functional networks themselves, and you already went through like the methods of how we identify them, but I am curious about how they got decided, how they were named. So who decided, and why are they named what they're named?
That's a great question. So I would like to start out in answering that question by saying that there's a lot of differences of opinion about what they ought to be named. I'm just gonna, where'd I put it? I had a figure in here that I took out, put something else on top of it. Yeah. I'm gonna show you this real quick. So first off, some of the networks that's a hard question, Sarah. So first off, we are in the relatively early days of functional MRI, right? 20, 30 years is how long we've known about MRI and been able to measure functional networks in the way that I'm describing today. We've had more than 100 years of neuropsychological literature in general, especially the kind of literature where you can look at stroke symptoms and look at what kinds of lesions give rise to what kinds of behaviors. So we've been defining networks based on what kinds, like what groupings of lesions give rise to, different behaviors for a long time. So for example, the language network, which includes what was once called Broca's area and Wernicke's, though that has been called the language network for a long time, it's quite reasonably called the language network. There's also motor networks, which include motor cortex and other regions that are premotor cortex that are involved in movement. Similarly, beautiful name, and it works as a name. More recently, so visual network has the same property, right? We can understand vision through the visual network. So there's been some sort of definitional regions, networks that have been defined based on their functions. More recently, though, people have used data-driven techniques to try to take information that comes directly from the brain and subdivide these brain networks, subdivide this information from the brain into separable networks. So for that kind of approach, they're trying to sit back and say, we're not going to use what we think we know about how the brain works in order to, put this idea on, on what we think is happening in the brain. So for example, the language so what they do is they use approaches like you might have heard of independent components analysis or principal components analysis. They use these kinds of machine learning type approaches to separate different parts of the brain into into regions that tend to be active together and regions that tend not to be active together. So I showed you the data showing the default mode network tends to be very tightly coupled to other parts of what we call the default mode network. So a couple of different people, not a couple, dozens of people have created these different networks based on large amounts of MRI data. And when they've done that, they've come up with things that look pretty similar to each other. So, for example, this is an image from, on the right is one of those people is named Thomas Yao. And on the left, another of those people is called Jonathan Power. And so each of them came out with a sort of a network definition. And I think, yeah, and they're using different data here, and yet they get very similar looking networks. So you can see, we're calling them, forget why for a minute, cinguloopercular network in purple and frontoparietal network in the sort of orangey yellowish, right? So no matter who does this separation, we're getting very similar results. And you'll notice we're looking at the left cortex here, right? Correct? And so some of these regions, they're language network regions, right? But we're not going to call them the language network because this has nothing to do with language. We're defining this just based on their functional connection properties. So instead of calling it like the language network, because it also includes some regions that got nothing to do with language, we are going to call it based on its anatomy. And so they're just going to call these regions, these networks, names based on their anatomy. So this is in purple, the cinguloopercular network. So the operculum is that space that is between the temporal lobe and the frontal and parietal lobes. Get in there, that's the operculum. The frontal part of the operculum is right here, and it's the middle of this purple blob. And the cingulate, this is the cingulate gyrus right here, and this is the anterior portion of the cingulate gyrus. So again, based on the anatomy. Frontoparietal, based on the anatomy, lots of frontal, lots of parietal involvement. So the more recent network definitions have sort of been mostly defined based on their anatomy, because we want to step back, or scientists have generally not wanted to make assumptions about what functions these brain regions serve before we have completely measured that.
Yeah, I think
Does that answer your question?
Yeah. So the ones where we're like visuals, like we're very sure about this.
We feel pretty confident.
We don't call it something like the occipital network or something like that.
I mean, we could. I honestly think it probably would be better if we did, because, well, there's people, there's lots of activity in the occipital lobe of people who are blind. Yeah. So is that the visual network?
Yeah, very true. Actually, if you could do the figure that's on top of this one. So as far as like translation to clinical world, we, as you were talking about, we see people who have domains in maybe some select places, but also more widespread. And this, could be part of the contributing is not only just damage overall, but these key places. And so I was wondering if you could kind of help us understand if we, have someone who we see the kind of patterns of widespread cognitive problems, then how could we use networks and the neuroscience around networks? Maybe in the future we'll have fMRI with our patients, but not quite yet, not with everyone at least. And so how could we use this to help us inform maybe stuff like recovery or what kind of declines we would expect from which hubs, that sort of thing?
Absolutely. I mean, I think as you kind of alluded to, I think the jury is still out a little bit on this because this is new kinds of data, but the idea is beautiful because knowing whether the damage, like before you do a single neuropsych test, the subject is, still passed out, right? Or after some insult, right? You should potentially be able to predict. I mean, we can already predict, for example, whether they're going to have language deficits or vision deficits based on a certain lesion. But we should be able to predict whether there's going to be involvement of multiple domains or not, based on whether the damage happens in a hub or not. And the idea is, if the damage happens in a hub, you are going to want to have the kinds of interventions that can be really across the board, that are gonna like integrate lots of different types of task processes all together. Whereas if you have someone who has a single domain deficit, you might be able to, for example, have interventions that just test that domain, or alternatively, interventions that sort of bypass it, right? So if they're having problems with, a particular, with orientation, you might, give them tools to try to use a strategy that doesn't require that. And so you might be able to use more strategy-based interventions for these patients, whereas it's unlikely that those are going to work as well here because you're hitting a lot of different domains.
So are hubs any more susceptible or recoverable than other regions?
I am not sure about the answer to that question. I think that's a really, really important question. And it's only been looked at very briefly in the literature, very, very little in the literature. If you think from an evolutionary perspective, you would think that the hubs would be less susceptible from like a blood flow sort of perspective.
That's what I was thinking about.
Yeah, that's what, that's what people are thinking. But I haven't seen anyone like systematically look at that. But I think that
maybe they will soon.
Maybe they will. Maybe you will. Maybe someone on this call will. Yeah.
Also, we just have a few minutes left, but I think it's, I know actually that you have a lot of great resources for that people can turn to learn more about networks and how we determine them and how they can be useful. So could you kind of lay some of those out for our listeners so they could follow up from this talk?
Sure. Are you talking about online resources, or
it can be textbooks or books, online resources, whatever you think is good.
That's good. So there's a really good, so this paper, the paper that this came out from, this figure came from, is from Katarina Grattan's lab, or she was in Steve Peterson's lab at the time, but Katarina Grattan, Gratton, 2012. And I thought I had put that here, but it's right here. That's a really good paper about this particular, I mean, it's what I'm citing here. And there's several really good books about this. One that you might enjoy is a book by Olaf Sporns called, I think it's Networks and the Brain. It's written in a very, relatively easy to read style, and it's about this general topic. There are also some really wonderful YouTube videos from a variety of different researchers who've looked at this, and I would love if I could point to a particular website that had them compiled. Maybe I'll put that on my website sometime soon, but I don't have them compiled right now. It's good.
Yeah, I can confirm that that book is really good. A good introduction and holds your hand pretty well,
Yeah.
Especially if you're not familiar with it. And I know that you do work in plasticity. And that's obviously something that we care about. We will hopefully can help our patients adapt. And so can you tell us a little bit about how the brain adapts to issues that happen in networks?
Absolutely. So that's a current direction of research. And there are two kinds of things that insults that could happen to the network, right? Well, I'm going to subdivide them into two, but one is a lesion, right? We could lesion a brain region. Another is a sensory change. So a change in experience in some way. And my lab studies changes in experience specifically due to vision loss. That's one of the things that we study. We also study aging, but with vision loss, what we find is there is an overall maintenance of these networks. These networks are still there. They're strong. They're viable in people with vision loss, but there are small modifications in the connection patterns that we think allow people to make better use of their remaining vision. So the idea is if you lose central vision, you start to use peripheral vision a lot. You fire together and wire together. I see anonymous attendee has that phrase. So you're firing, you're activating these regions that process information from the peripheral vision. And what we're finding in new data that we is not published yet, but what we're finding is that these regions that you're using more are actually increasing their connections to some of the brain regions that we know that they're going to that have functions that we know people with macular degeneration need to use on a daily basis. So for example, MT is a region that processes moving stimuli, including looming stimuli, like when you walk towards a doorway, the sides of the door loom towards you. And we find that the regions that process peripheral vision tend to upregulate their connections to this area MT in those patients with macular degeneration. So that's one of them. The other thing I skipped is, if you have a lesion, that's another way that you can change the system. And so you can change some of the functional connections around that lesion. We think that that's adaptive, helping you to your brain develops strategies to get around the use of the lesioned region. That's the current thinking.
Yeah, awesome. Brains are amazing. They can recover from a lot.
Very true.
All right. Thank you so much. That's all the time that we have for today, but thank you all for joining us. And again, this is the last lecture in the summer neuroanatomy series, but we will be starting up another volume of the neuropsychology. So be looking out for that. We'll be emailing out to our current subscribers, but if you aren't, make sure that you subscribe to our emails and also follow us on Twitter and look at the YouTube if you want to catch up on any of the neuroanatomy series. So thank you, everyone, and have a good rest of your day. Okay.